Okay, so our special topic at the show this year is about serpentine habitats. And we chose this topic because the serpentine flora is a really important part of California's botanical diversity. And because we here in Humboldt County have lots of good examples of it. It's close enough that we can go see it. And some of us do. We really enjoy it. It's one of our favorite places to go. And then a lot of us know the term serpentine. We know Horse Mountain is a serpentine habitat, but we don't really know what that means. You know, okay, it's sort of barren rock, but we thought it would be good to learn what it really means and what it means for plants trying to live on it. And that's what our speakers are helping us do. And tonight's speaker, well, last night's speaker was a geologist telling us about it. And he was a geologist who knew something about plants. He put in plants too. Tonight, we're going to have a botanist who I bet knows a lot of geology. And we'll see what he tells us about it. And Nishanta Rajakaruna is here tonight because when I wanted serpentine speakers, I sent a message to Nick Jensen at the state CMPS. I thought there might be somebody at the state who could talk about serpentine habitats and how they're important for a flora. Well, Nick said he couldn't do it, but he recommended Susan Harrison and Nishi Raja, darn, and Nishi Rajakaruna. There, got it. Well, I didn't know this guy, Nishi. So of course I asked Google, what do you know about this guy? And Google took me straight to the Geoecology Lab of the Polytechnic State University, San Luis Obispo, which we call Cal Poly. However, when Humboldt State becomes a polytechnic, somebody's going to have to add to their name.
There'll be 3 then.
Yeah. Okay. Okay, and here's what I found on the homepage of the Geoecology Lab. It said, we are a bunch of biologists interested in plants, lichens, microbes, and other organisms associated with rock outcrops in California and beyond. We love hanging out on rocks. The more harsh or extreme they are, the more we love them. We also love hanging out at dumps. And that was good enough for me. I decided these people are some we want to hear from. So I wrote to Nishi, and gosh, he must not be very busy because he wrote right back and said he'd be happy to speak to us. And I'm really pleased that he's here tonight. So Nishi, take it away with your screen share.
Got it. Well, thank you so much, Carol. It's indeed a pleasure meeting you all. And I always love meeting with CNPS crowds. They're the best among all audiences I generally speak to. They're passionate about plants. They like hiking. And so they're my kind of people. So I never say no to a CNPS invitation. So thank you for the introduction as well. And thank you to all 16, actually 17 with Leonard in the back of that screen. For spending the Sunday evening with me, talking and listening to me talk about why I think serpentine plants are cool model systems to ask all sorts of cool basic and applied research questions. So I'm going to take you on a kind of a brief world tour of places I've been to that has amazing serpentine floras and also lichens. And kind of share with you some of the cool research, both basic research and applied research that is happening from California all the way to places like New Caledonia, Malaysia, and Albania. So hopefully you'll get a kind of a feel for where serpentines are around the world and how exciting they are for people who are studying them. So with that, let me start my presentation. If there's anything that's unclear, I mean, I'm perfectly fine with you raising your hand, and hopefully Carol or I will see it and we can try to respond to it if it is a short question. But if you have longer questions, hold on to them. We can discuss them towards the end of the presentation. So I've had a really long-term fascination with geobotany, and a lot of my research and teaching over the years have used this model as these habitats of rock outcrops. serpentine and other types of rock outcrops as kind of model settings to ask questions about ecology, about evolution, and also to practice things in conservation biology and restoration practices. So my fascination with geobotany started pretty early in my academic training. I was an undergrad at a place called College of the Atlantic in Maine. And during my senior year, while taking a plant systematics class, I came across two papers written by Professor Art Krukeberg, who was then a professor of botany at University of Washington, a LA resident, a California native, but worked at University of Washington for close to 50 years, and someone that we consider to be the father of geobotany in Western North America. So after reading these two articles as a 21 or 22-year-old, I think there was no turning back. I fell in love with serpentine even before I had stepped on a serpentine outcrop. I'd never, ever seen one, but his writing got me hooked. Art has written a lot of papers and books on geobotany, and I'm sure some of you guys would have encountered some of these papers and books that he wrote along the way. He did a lot of cool experimental work starting in the 1950s all the way up until about the late 90s. He was active as a serpentine ecology researcher, and his books continue to educate people across California and the world about why rock outcrop plant communities are really exciting habitats to study diversity, ecology, evolution, and also think about the importance of conserving rare plants and plants that are endemic to certain parts of the world. So I got to know Art when I was a graduate student at University of British Columbia. I think it was a great kind of honor and a privilege to get to know him. I spent a lot of time in the Siskiyous with him in the late 90s and early 2000s. I even TA'd a geobotany class at the Siskiyou Field Institute for Art Krukeberg in the late 90s. And also had the honor of writing his in memoriam a few years ago when Art passed on at the age of 96. A really productive life, an amazing person, and a wonderful geobotanist, and someone that I consider to be a great mentor and somebody who got me hooked on serpentine even before I was able to see an outcrop. So over the last 17 years, I've taught at College of the Atlantic in Maine, which is where I did my undergrad. I've taught at San Jose State. And since 2017, I've been teaching at Cal Poly in San Luis Obispo. And I've often used these outcrops, whether it's serpentine or dolomite or gypsum or granite or sandstone, these kind of rock outcrop habitats as model settings to do my classes, teaching, as well as to do research. And while I was at College of the Atlantic, I taught a class called Plants with Metal, metal meaning courage, not necessarily metal, like a heavy metal, but it was about plants that grow on metal contaminated sites. And it was a plant ecophysiology class. And I basically used some of the mines that we had in Maine, copper and zinc mines, as a setup, as a setting to teach basic botany and things like ecology and physiology and even habitat restoration by using that model setting. At Cal Poly, I have a class called Geoecology, which is a class that kind of discusses the importance of landforms and lithologies, parent material, in terms of shaping diversity of plants and other organisms, both here in California as well as around the world. So before I get to serpentine, I kind of want to talk to you about what I consider to be this kind of weird or harsh soil, right? So what do I really mean by a harsh soil? So here's a serpentine outcrop in Sri Lanka, which is where I'm from, that I've done some work at. And you could see this really sharply defined boundary between non-serpentine, more kind of sandstone type soil, and serpentine, which is really red over here. So when I talk about these harsh soils, I'm really talking about soils that have too much or too little of something that is essential for plant growth, right? So if you have too much nitrogen or too much phosphorus, things that plants require, but too much of it can lead to toxicity. And then if you have too little of it, right, like serpentine outcrops generally have very little nitrogen and phosphorus, and that can lead to deficiencies. So plants either deal with toxicities or deficiencies of essential nutrients. Harsh soils can also be soils that have toxic elements that plants don't require for growth, right? Things like chromium, cadmium, cobalt, mercury. Plants don't need them, but if plants are growing in soil that have those elements, that could lead to toxicity as well, right? So that could be another way to define a harsh soil. Finally, harsh soils can be soils that have really poor structure or texture, right? Soils that are very rocky or sandy or have too much clay can be hard medium for roots to grow through. And those kinds of soils can be harsh as well. So the harshest of the harshest soils will have some combination of all of these three factors that are important in terms of defining a harsh substrate for plant growth. Right? So when plants encounter these harsh substrates, like this setting that I have on the screen, a serpentine outcrop in the southern part of Sri Lanka, where you have a really sharply defined non-serpentine and a serpentine boundary. So plants can exclude it, like plants can just avoid colonization, or even if they colonize it, they're not going to make it, right? So these are what we call the excluders or the intolerant species. Not every species in California can grow on serpentine soil, right? So the ones that do not grow on serpentine soil are the ones that are excluded from it, right? They're intolerant. You have then what we call the indifferent taxa. These are taxa or species that have broad substrate tolerance. They can grow on and off of serpentine. So think about something like Heteromeles, Toyon, or Ceanothus, right? There are Ceanothus cuneatus. There are species that can grow on and off as if there's no difference in the substrates that broadly tolerant in terms of the harshness that the serpentine soils bring about, as well as the other substrates that they grow on. Then, of course, so these are the ones that Art Kruckerberg used to call Bodenwag taxa. These are the soil wanderers. They can tolerate broadly different types of soil conditions. Then there are the endemics, the ones that are largely restricted to one type of substrate or another, right? So serpentine endemics are the ones that only grow on serpentine soils or largely restricted to serpentine soils. So this is how generally, whether it's serpentine or whether it's limestone or whether it's gypsum or dolomite, plants respond to these kinds of harsh substrates. So, people have been studying kind of plant tolerance to unusual substrates for a long time. And what we generally find is that there's a lot of variation, genetic variation for tolerance to different types of substrates. So what you see here are two lineages or genetic lineages of Arabidopsis species that has serpentine tolerant populations that were collected from serpentine soil and populations that were collected from non-serpentine soil. Right? So you bring the seed into the greenhouse, you grow them on serpentine soil. When you grow serpentine origin plants in serpentine soil, they do perfectly fine, regardless of where they came from, right? They are tolerant of serpentine soil. When you grow the non-serpentine populations of the same species, they look identical morphologically, but you grow them on serpentine soil, they don't do well, right? So that shows that there is genetic variation even within species for serpentine tolerance. There are serpentine tolerant genotypes or populations that are locally adapted to serpentine soils. And we see these kinds of populations all over the place here in California, as well as in other places. And you can think of these populations as the kind of the starting or the stepping stone to a potentially a new species down the line, right? They are distinct enough physiologically that over evolutionary time, they could become a serpentine endemic species or a serpentine tolerant species. So my research happens mostly in California, but I also spend quite a bit of time working in the Northeast. Most Californians do not realize that there's a lot of serpentine in Eastern North America as well. And I'm going to talk about that later, but I study some of the serpentine outcrops in Maine and Quebec and Vermont. I also spend quite a bit of time in the Southern Urals in Russia, working with Russian colleagues on serpentine lichens. And then I spend time in South Africa, where there is quite a bit of serpentine, almost as much serpentine as California has, and a lot of endemics, including some really cool plants that I'm going to talk about later on in the presentation. And then, of course, I'm from Sri Lanka. I grew up in Sri Lanka, and I have been working on Sri Lankan serpentine outcrops over the last 20 years as well, and have discovered some interesting plants that are found nowhere else. So I'll take you through to all of these countries as well as to some other places that I think are really interesting to discuss. So climate, we all know that climate is really important in terms of understanding distribution patterns. We know why certain things are where they are because they are, either tolerant of or intolerant of a particular climate. But within a climatic zone, right, if you think about the Mediterranean climate or a temperate climate or a tropical climate, within that climatic zone, geology is really, really important in terms of kind of generating habitat diversity and kind of leading to really cool species distribution patterns. So when I was a graduate student at University of British Columbia, I would drive down to Jasper Ridge Biological Preserve, which is Stanford's biological preserve in San Mateo. And this is what this site looked like in the 90s and 80s and 90s. So what you see here is the serpentine outcrop at Jasper Ridge at Stanford, sharply demarcated by a sandstone outcrop. You can see the sandstone serpentinite boundary right where the grasses end and the gold fields, the Leycinia californica and Leycinia gracilis species start. So this is really interesting because this population, Leycinias are annuals, right? They go from seed to seed within 3 months. But every year they germinate, and by the time they go into flower, they maintain this really sharply defined boundary. So when I was a graduate student, I did some what we call reciprocal transplant experiments. I mean, this is a cool tool we botanists have to move things around. I mean, plants root, right? It's not like trying to move an animal from one habitat to another and to see whether the animal is going to be adapted to that new habitat. In a plant, when you plant it, it's going to be there, right? So you can do this, what we call reciprocal transplant experiments. And when we do that, we saw that the Leycinias cannot really compete with the grasses on the sandstone. They get outcompeted, outshaded. So they don't really get to the flowering stage in a grass thick sandstone outcrop. When you move some of the grasses, especially the non-native grasses, to the serpentine outcrop, they don't make it. I mean, they don't even reach the flowering stage because they're intolerant of some of the toxicities and the nutrient poor conditions that the serpentine soils have. So by doing this very simple experiment, by moving things from one habitat to another, right across the boundary, we were able to show that the soil conditions are really important in terms of maintaining this really unique pattern that you find at Jasper Ridge as well as in other places. So, I think of the world as a kind of a patchwork quilt, right? I think of this kind of surface of the earth covered with all sorts of different geologies, right? There's granite, basalt, serpentine, sandstone, shale, side by side. And when you have these kinds of, sharply defined little outcrops that are scattered all over the surface of the earth, you find that different plants that would be locally adapted, like the Leycinias we just saw on the serpentine outcrop, are better fit or better adapted to grow on serpentine. So when you have these different species that are locally adapted to one type of substrate or another, over time, what you end up getting is different plant communities. You get plant communities that are distinct to one type of geology or another, right? Even at Carrizo Plain, with the far left photo here, we know that these patches correspond to levels of salinity in the soil, right? So some of these species are better tolerant of high salt conditions, high clay content, and there seems to be a really strong correlation with soil chemistry and soil physical conditions associated with this distinct patches of color that we define as a superbloom, right? There's serpentine, there's all sorts of other types of outcrops that generate really cool, interesting looking plant communities. So while we're going to talk about serpentine today, I also wanted to acknowledge that there are different types of substrates all across California that have interesting floras, right? Serpentine has received a lot of attention because people like Art Kruckeberg, Susan Harrison, have been using serpentine as a model system for a long time. But there are things like limestones, dolomites, and gypsum that have really high levels of calcium and sulfur that can also be toxic to plants. So they also generate really unusual plant communities. You have serpentinite, of course, that we're going to talk about today. And if you went to the geology talk, I'm sure you heard that Serpentinites have high levels of magnesium, high levels of heavy metals, low calcium, low nitrogen, and low phosphorus. There are mine tailings all across California: quicksilver, gold mining, nickel, asbestos mines. These are disturbed settings that have really harsh soil conditions. So there are plants that have adapted to deal with these conditions as well. Another interesting substrate that I'm interested in, that I've been working on, are guano deposits. These are settings where seabirds nest, and where seabirds nest, the soils have high levels of nitrogen and phosphorus, and soils can be very acidic. I mean, some of the seabird nesting sites have soils with pH of about 3.5, which is very, very harsh type of substrate for plants to grow in. And we know that Leucaena maritima, which grows along coastal bluffs and on Farallon Islands of the coast of California, is an endemic to guano soils, right? So there are other substrates that are interesting as well. So geology has really contributed to California's plant diversity, like Carol pointed out. We of course point to serpentine as the primary geological factor, but of course there are other types of substrates that I just talked about. If you look at all of California's endemic plants, about half of them are substrate specialists. Right? I mean, that is a pretty high number, right? Showing that, yeah, climate is important, aspect is important, inland coastal gradients are important, but really elevational gradients are important, but geology also contributes greatly to endemism and diversity in California. And of course, if you look at rare plants listed that has California rare plant rank that CNPS uses to list their rare plants, about 50% of rare listed plants by CNPS are also substrate specialists, right? So this is some work that Nick Jensen and I are doing to kind of show the importance of geology to California's both endemism, diversity in terms of endemics, as well as diversity in terms of rare and endangered species. So geology is important. So any kind of plant conservation or habitat restoration effort we get involved in, we'll have to pay attention to substrate plant associations. Because if you're trying to restore a landscape, right, you're trying to restore a serpentine outcrop, you don't want seed from a sandstone outcrop. You want seed from a serpentine outcrop, because those are the seeds that are going to be successful in establishing a population on a serpentine outcrop. You got to pay attention to whether they're from a north-facing slope or a south-facing slope, right? The reason that a lot of our restoration efforts fail is because we don't really pay attention to exactly where the seeds come from. So it's really, really important to think about substrate associations as fundamental to thinking about conservation and restoration efforts. So quickly, just because this talk is going to be on serpentine, just letting you know that there are lots of other cool plants across California that we think are largely restricted to different types of substrates. So these are what we would call endemics that are understudied, right? The laterites that have Eriogonum and Manzanita species and a Polygonum species endemic to it. There's dolomite endemics, gypsum endemics, granite endemics, gabbro. These are species that have been largely collected from these types of substrates across California, but very few people study them, right? So it's really important to think about moving beyond serpentine at some point and appreciating all of the different types of geologic substrates that California has, and trying to kind of do the types of work that we've already done on serpentine on these understudied habitats. But of course, serpentine is the king of all types of substrates. Everybody loves serpentine. And there have been a lot of books written on serpentine outcrops, starting from the 1984 book by Art Krukeberg. There have been books written by Europeans. And the last book that you see on the far right is a book that Susan Harrison and I wrote called Serpentine, Evolution and Ecology of a Model System. And basically kind of promoting serpentine as a model habitat for research for people who haven't really thought about serpentine as a cool habitat, right? So a lot of books that have come out on serpentine soils. So I know you guys went to the serpentine talk yesterday, so I don't want to repeat it, but let me ask you a question. Is serpentine a rock, a soil, or a group of minerals? Anybody?
Group of minerals.
It's a group of minerals, right? I mean, I have a lot of geology friends and soil scientists who hate botanists for calling everything serpentine, whether it's soil or plants or lichens, anything that's found on a serpentinite outcrop, we loosely use that word serpentine to refer to, right? But generally, serpentinite is the rock. Serpentine is a group of minerals, but botanists have basically kind of very loosely used it over the years. Serpentine is found all along continental margins, wherever there is mountain building activity, mid-oceanic ridges, and the soils that are weathered from these rocks that have high iron and magnesium are called serpentine soils. They're high in heavy metals, nutrient imbalances, nitrogen is low, phosphorus is generally low. What's really important, we think, is the calcium to magnesium ratio. Calcium is really important for plant cell development, cell wall development, and calcium is very limiting in serpentine rocks, in serpentine soils, because the way the serpentine forms leaches out calcium while the process is happening. So serpentine soils have really limited amount of calcium in it, and that can cause a lot of deficiencies for plants that are growing on serpentine soils. And as you know, in California, serpentine outcrops are generally rocky, dry, open, so they are really, you know, exposed to the elements, and all of these harsh chemical and physical conditions lead to these distinct plant communities that are very high in plant endemism, meaning high restriction, and also lots of rare plants, right? So, you know, Californians don't realize that, you know, I mean, not just Californians, I mean, because we think about serpentine as our rock, which it is, it's California state rock, serpentinite is, but there's serpentine all over the world. I mean, there are serpentines all along every continental margin. So the blue circles and the green triangles are all serpentines, and they are found all over the world. I mean, they only cover about the % of the surface, but there are little outcrops all across the world. Wherever there's massive tectonic activity, you can get chunks of ultramafics or mantle rocks that have gotten exposed to the surface of the earth. If they had gone through the process of serpentinization, you end up getting rocks that would eventually give rise to serpentine soil, right? So they're everywhere, not just here in California. And serpentinites are also important in our lives. I don't know whether the geology, I missed the talk, I'm sorry about that, whether he talked about this, but asbestos comes from serpentinite rocks, right? And stainless steel, nickel chromium that we used in stainless steel, if you've ever touched a stainless steel item, you're looking at, you know, a product of a serpentine rock. And if you travel in places like Cuba, South Africa, parts of Russia, you see people selling jewelry and statues and all sorts of little, you know, crafts made out of serpentine rocks because it's easy to work with, and it has a really nice green shine to it. So they are important in our lives in other ways beyond botany as well. And in fact, there are people who think that serpentinization process that I'm sure the geologists talked about, the whole hydrothermal alteration of these mantle rocks that gives rise to the parent rocks of serpentine, is thought to be kind of a place that early life may have evolved, right? So there's this idea that the hydrothermal alteration or the serpentinization process, which generates serpentine rock, generates enough heat and energy, raw material, for chemosynthetic microbes that require chemicals to thrive on, right? So in fact, you know, that's why NASA is interested in serpentine settings, because they look at these actively serpentinizing areas to look for like life forms that might represent early forms of life that we might find in places like Mars, right? So there's a lot of interest in terms of serpentine as an early setting to understand early life. If that's not enough, all you need to do is run into a person down the street who pulls up the sleeve and shows me the chemical formula for serpentine tattooed on her arm. So this is a grad student, Daniel Black from UC Santa Barbara, that I ran into at a California Botanical Society meeting. And she comes up to me, knowing that I work on serpentine plants, pulls over the sleeve and shows me her serpentine tattoo, right? So I guess it makes a great tattoo as well for people who love serpentine so much that they actually want it tattooed on their body. So some of the cool places that I want to kind of take you to are New Caledonia, where probably the best place to be if you are a serpentine enthusiast. 83% of New Caledonian endemic plants are found only on serpentine soil. Cuba is another place that I've been to, and Cuba is a fantastic set of islands to look for serpentine endemics. A third of serpentine species endemic to Cuba are endemic to serpentine soil. And lately, my colleagues have been working in Malaysia on the island of Borneo and finding that at least 5% of the total vascular flora of Borneo is endemic to serpentine soil. So serpentine soils are found everywhere. And I would put these three places as kind of the most diverse and most interesting places for serpentine botanists to go to, because you're going to find a ton of endemic species, including plants that are found only in that particular country, and they are entire families that are endemic to serpentine soil. So New Caledonia is one such place. The island of New Caledonia is to the north and east of Australia, for people who haven't thought about that part of the Pacific before. A chain of islands, and serpentine covers a large part of the landmass. There's a lot of nickel mining that is going on, and that has caused a lot of problems in terms of creating very devastated habitats and lots of rare plants. Serpentine outcrops are found, rare rainforests growing on serpentine, really ancient serpentine soils. There are rock outcrops, savanna, chaparral type outcrops. So it's very, very diverse. What's really cool about New Caledonia, there are 20 genera in New Caledonia that has only one species in it, and they are ultramafic or serpentine endemic. It's the only country in the world that there are families that are endemic to serpentine soil. There are 8 families in New Caledonia, plant families found nowhere else in the world. Each family has only one genus in it, and all those families are endemic to serpentine soils. And it's an amazing place to be if you are interested in serpentine endemic plants. I was in Cuba with the giants of serpentine plants in California, in I guess in the North America. Art Kruckerberg on the left, Richard Walker, who was also a University of Washington professor, who studied the calcium to magnesium ratio. I mean, he did experiments in the 1940s looking at why calcium to magnesium ratio was so critical for serpentine tolerance. Bob Coleman, who just passed on this year, a Stanford geologist who is probably the world's leader in understanding how serpentine outcrops form in mid-oceanic ridges. And Earl Alexander, who some of you may have met, who's the only person alive other than me in this photo. He's a serpentine soil scientist who has mapped much of serpentine soils, including those soils that of North Coast County, like in the Siskiyou region. And Earl has been very active since the 1960s and has done a lot of work in California. So I was lucky enough to be among these giants of serpentine ecology in Cuba in 2003. I also ended up going to Mount Kinabalu in 2014 in Borneo, a really hotspot for serpentine endemics. And this is Bob Boyd in the middle, and a former student of mine, Tanner, who's an environmental consultant now. Bob Boyd, I'm going to talk about Bob's research later on. He studies Streptanthus Polygloidis, which is a serpentine endemic in California, and I'll talk about his research a little later. But Borneo is amazing. I mean, Sabah has all of these orchids that are endemic to serpentine soil. They are really visually striking species. I mean, these are just a handful that I'm showing, but there are lots and lots of serpentine endemic orchids. Some are epiphytic, some grow directly on the rock, some grow on the understory of other plants. Really, really rich rainforest-like serpentine settings. There are pitcher plants that are just unbelievably unbelievable in the way they look. Nepenthes raja is about a foot and a half long. And there are lots of these pitcher plants that are hanging from trees or growing directly on the rock. And if you're a pitcher plant, I mean, I guess the serpentine outcrops are not a bad habitat to grow on, because you can trap insects and supplement your nitrogen requirement rather than trying to rely solely on the soil nitrogen. You can get nitrogen from insects. So pitcher plants are found everywhere. And Mount Kinabalu about 5,000 meters, 4,000, 5,000 meters. So it's pretty high up, and these pitcher plants are found at different elevational ranges across this mountain range. Not all of Mount Kinabalu is serpentinite, but much of it is. Of course, then coming back to California, California is obviously an interesting place, as you guys know. Only 1.5% of California state is covered by serpentine rocks, but 14.7% of California endemics, which are about 255 taxa, are endemic to serpentine soil, right? So serpentinite is, of course, California state rock. Even though it is stated as serpentine, technically it should be serpentinite, is what should be California state rock, but really well deserving of its naming as the state rock because it contributes to diversity. Economically, it's been important. Gold is associated with serpentinite. Quicksilver, mercury is associated with serpentinite. Asbestos, nickel, chromium mining. So economically, for early California, serpentine has also been really important. We talk about plants, but we forget there are other organisms that are associated with serpentine outcrops, and the world's first serpentine endemic vertebrate was described recently from the Red Hills of Tuolumne County in California. So this is a fish species that seems to be found only on little rock pools that are associated with serpentinite bedrock. Right. So, I mean, it's hard to describe endemism in a fish because you assume that the fish is going to keep swimming. But apparently these authors say that this species, every time you find this California roach, it's called the Red Hills roach, is found only on these serpentine type bedrock pools in that particular county. In California, you guys are familiar with these guys. They're found in Northern California. You've seen the Darlingtonias that grow on serpentine fens. You've seen the California lady slipper, the Cypripedium californicum, that are also found, not always, always, but often on serpentine fens. And these were some of those kind of striking, charismatic species. The Cobra Lilies are just a wonderful plant to find on a hike. And, these are some of the plants that bring me to the North Coast, whenever I have the time. In San Luis Obispo, where I'm currently speaking from, we have a bunch of endemics, including several that are named Obispoensis, meaning that the type specimen was collected from San Luis Obispo. We have a Toadledia calicota species, the Manzanita. We have the Sargent cypress. We have the Carex species. I mean, these are rare endangered species that have their kind of prime range in San Luis Obispo County, including a Cirsium species, a bog thistle species that is found on some of the seeps on serpentine outcrops in San Luis Obispo. So a lot of cool plants in San Luis Obispo. So that's why I'm really excited that I'm actually here, because I can just walk out of my apartment and within 10, 15 minutes, I'm on a serpentine outcrop with lots of cool California endemic serpentine plants. Recently, Dave Keel, who's our senior botanist, now retired but still pretty active, described a new species called Corysanthe aphanantha from Irish Hills. I don't know whether you've been to Irish Hills, but it's another really cool place to go if you're interested in serpentine plants on the Central Coast. And Corysanthe aphanantha in the Polygonaceae is closely related to Corysanthe breweri, which is found sometimes side by side. But the primary difference is in the inflorescence. The flower has some cool features that distinguish it from the Corysanthe breweri, the more common serpentine endemic. They're both serpentine endemics, but they are sometimes found together and sometimes found in different types of serpentine soils. So I have an undergraduate student studying these two plants, kind of looking at the subtle differences in their habitat. And we found that Corysanthe aphanantha, the one that is recently described, is generally found on serpentine soils that have higher organic matter content, higher cation exchange capacity, soils that are much better for growing. So among serpentine, there's better serpentine and harsher serpentine. So this guy seems to favor the better kind of medium of serpentine and also lower pH serpentine soils than the high pH serpentine soils. So there are subtle differences in the ecology, even though they are both found on serpentine soil. So this was recently described in 2019 by Dave Keel and Kristen Nelson, who's the vice president of the CNPS chapter of the San Luis Obispo chapter of the CNPS. Dave Keel also recently described a new monkey flower from Irish Hills. It's Erythranthus serpentinicola, which has much smaller flowers than the Erythranthus guttata, the sheep monkey flower, which sometimes grows nearby. But this guy grows directly on the rock. So Erythranthus serpentinicola grows directly on the serpentine rock. It has, we think, has an annual growth habit, whereas the Mimulus guttata or the Erythranthus guttata has more of a perennial growth habit. So, in terms of California serpentine flora, a lot of the endemics are found where you guys are, right? So the endemism is much higher on the north coast and the Klamath Mountain ranges. And Susan Harrison's research shows that it's because there's higher precipitation in the north coast. There are much greater extent of serpentine outcrops, like in the Siskiyou region and some of the northern outcrops, and also generally long exposure of rock to the Earth's surface. These factors may have led to greater levels of endemism in the northern parts of California compared to the southern coast. But even, like I said earlier, like even on the central coast, wherever you get serpentine, the more you look, the more you find interesting species. So that's kind of a quick overview of why California is cool, along with places like New Caledonia and Cuba. But I always try to tell people that there's some really cool serpentines on the East Coast, right? People, I don't know how many of you are from the East Coast, whether you came from the East Coast, or whether you've been to some of these places in Quebec and Newfoundland and parts of Maine and Vermont. But if you think about the total amount of exposed serpentine, there's as much serpentine on the East Coast as you get on the West Coast, right? The only difference is that the East Coast serpentines were glaciated, right? So there was ice covering these serpentine outcrops for a much, much longer time, and up until about 10,000 years ago. So they haven't had much time for evolution to take place, maybe like 10,000 years compared to millions of years that places like California would have had ice free. So a lot of serpentine outcrops all across the East Coast, along the kind of the Appalachian mountain belt, going from Newfoundland down to Georgia and Alabama. There are endemics that very few people know about. I mean, there are at least 6 endemics that we've described from the eastern part of North America, which includes a fern in the Adiantum group, Adiantum viridimontanum, which is a polyploid fern between 2 ferns that are found on the East Coast. There's a bunch of Caryophyllaceae, the carnation family. There's a Pachera, which is a Asteraceae plant, and another aster that is endemic to serpentine soil. So there's some interesting plants, not as many, not 255 like we have here, but there are some endemics in the East Coast as well. So, a lot of my research over the last 10, 15 years have been kind of targeted at understanding how these weird soil boundaries contribute to evolution of serpentine endemics. So we know that whenever you find a serpentine outcrop next to a sandstone outcrop, where the soils are generally benign and harsh on one side, benign on the other side, you have strong selection for genetic changes to take place. And over time, this can lead to serpentine tolerant genotypes, like we talked about earlier on, becoming, colonizing serpentine outcrops, and over evolutionary time, becoming new species. So a lot of California serpentine endemics are what we call new endemics, endemics that would have evolved over the last, 3-5 million years in response to exposure to some of these serpentine outcrops that have appeared on the surface. One of the projects that I did as a graduate student is at Jasper Ridge, where we documented 2 closely related species. Remember, I showed you this photo earlier, but I only talked about the serpentine sandstone boundary. But if you look at this photo closely, you see that there is a brighter yellow down here and a lighter yellow up on top. So what you get is 2 closely related Lascinia species, Lascinia gracilis and Lascinia californica, growing side by side, but they maintain a really sharp boundary between the 2 species. And over greenhouse studies that we've done, we've shown that Lascinia gracilis is a drought-tolerant species that seems to be able to deal with the drier serpentine soils on the upper reaches of the outcrop. And Lascinia californica, which is more kind of a moist-loving plant is able to deal with the higher ionic content that you find at the bottom swale. So they are, closely related species that are growing side by side, right? But they seem to know exactly where they're growing because they never really cross that boundary as much. And when we do these reciprocal transplant studies, we know that Californica cannot tolerate the drought upon the upper reaches of this outcrop. Gracilis cannot tolerate the harsh ionic conditions at the bottom. And mind you, this is only 40 meters from 60 meters from here to there, right? So it's not a long distance, but they have basically separated out their little niches, right? And they are found in places where they're best fit. And so this is one of the first studies that documented that you don't necessarily always have to look at serpentine, non-serpentine boundaries. You can also look at boundaries within a serpentine outcrop where soil heterogeneity due to aspect can potentially lead to the maintenance of different species, right? We've also shown that these species, even though they're closely related, have reduced reproductive compatibility. So there's a reduction in gene flow, even though there is some overlap in flowering, right? So generally, when you look at this outcrop, this is Californica, the brighter yellow. So those are flowering a little later than the ones that are found in the drier part of the outcrop. So these guys are already past early flowering when these guys go into flower, right? So there is a flowering time difference that reduces gene flow between these populations. When we bring them into the greenhouse, grow them under identical conditions, we find that they maintain this flowering time difference, right? Gracilis is always flowering early, about 7 days before Californica does. So by flowering at different times, plants can limit the amount of genes they exchange. And when you have limited gene exchange, obviously over time, you could evolve into a distinct biological entity because you're not exchanging genes with members of a nearby population. Right? We've also done crossing studies to show that Californica and Gracilis, even if you get them to flower at the same time, and if you cross them, hand cross them, there's limited pollination success and seed set when you cross A's versus C's, right? Within each species, there's greater pollination success, greater seed set than when you cross the two together. So even if they were to flower at the same time, you have a reduction in gene flow. So that would suggest that they are closely related. They grow side by side, but they know who they are. They're not interbreeding with the population that is side by next to them. Right? So these kinds of really cool evolutionary processes can be studied by looking at plants that grow on serpentine soils. So I'm going to move on to lichens very quickly. Lichens are really exciting, and very few people have looked at lichens on serpentine outcrops. And I'm sure you guys may not have looked at lichens, but lichens are also really cool. We looked at lichens in South Africa through a National Geographic Society-funded expedition. The first time people had looked at lichens in South Africa since 1957. And we were looking at lichen diversity along a climate gradient, right? And we found, collected over 1,000 specimens in 3 weeks across this kind of climate gradient on and off of serpentine soils. We have described 6 to 8 new species to science in that 3-week expedition, including an entire genus new to science. And this is not because we were good, we were great lichenologists. It was only because no one else had looked at these lichens on these types of outcrops. Right? So this paper just came out describing a new genus, and it's exciting. There's a lot to be done. I mean, we haven't even gotten through 10% of the collections we made, right? So we are doing a similar study here in California. Here's my grad student, Michael Mulroy, doing a similar lichen study along a coastal inland gradient here on the Central Coast.
Hi there, I'm Michael Mulroy. I'm a member of the Geoecology Lab at Cal Poly, headed by Dr. Nishanta Rajakaruna. I'm here at one of my ultramafic, aka serpentine sampling sites, looking at lichens. I'm studying Saxicolis lichens on sandstone and ultramafic substrates along a coast inland gradient in San Luis Obispo County and some surrounding areas. So really surprisingly little is known about these Saxicolis lichen communities worldwide, but especially in North America. So our goal is to better understand what factors, including maritime influence and substrate factors like the geochemistry, actually affect what types of species are growing where. To date, I've sampled 12 of 16 total sites, made approximately 500 lichen specimen collections, over 180 of which have been identified as species, and this represents approximately 90 distinct taxa so far. This includes over a dozen species newly recorded from ultramafic substrates in North America, as well as range extensions for several notable species.
So Michael's study is ongoing, but we have already found some really cool stuff and some things that we have sent to experts to identify. But again, lichens on ultramafic rocks or serpentinite rocks have been understudied, even in a place like California, where there's a lot of work being already done. In South Africa, we've been also looking at biocrusts. These are algae and cyanobacteria that grow on soil surface. And we've found 9 genera of algae and cyanoprokaryotes or blue-green algae that have never been reported from South African soil before. So again, this is not a lot of like extensive research. These are just soil samples we collected while we were on the go, right? We were basically collecting them whenever we saw an outcrop, and we are finding new species and new taxa because they have not been looked at in the past. So finally, I want to kind of talk to you about some really cool applications of the study of serpentine plants. I don't know how many people have heard of this group of plants called the metal hyperaccumulators of serpentine soil. Metal hyperaccumulators take more than 1.1% of their dry weight in a heavy metal. This happens to be about 1 milligram of metal per gram of dry leaf tissue. And this can be toxic to most organisms, including to us, right? So, but there are plants that do this. So the first plant that was documented to be a metal associated plant was by an Italian naturalist in the 1500s, where he wrote in his diary that if you find this Brassicaceae plant, it's a good place to mine for nickel, right? So this is 1583, right? People did not have the tools we currently have to look at how much nickel is in the rock, but they used plants to indicate rock that could potentially be good rock to mine for heavy metals. So this is a bioindicator or plant indicator of a serpentinite rock. We now know that this plant has about 2% of its dry weight in nickel, right? That's a huge concentration of nickel in the tissue of this plant, and it is an endemic to serpentine soils of Mediterranean Europe. The plant that is considered to be the kind of the king of metal hyperaccumulators comes from New Caledonia, where an accidental discovery led to this plant where they found 25% of their dry weight of the latex that seeps out of the trunk rich with nickel. Every part of the plant actually had nickel in it beyond the threshold that defines hyperaccumulation. So these are plants that are sucking up nickel from the soil, localizing and accumulating the nickel in the plant to levels that would kill any one of us who would even taste a plant like this. So here's my friend, Anthony van der End from the Netherlands, who is in Queensland right now, who is an expert on the study of metal hyperaccumulating plants.
Here in the jungle of Grande Terre, scientist Anthony van der End of the Australian University of Queensland is researching plants with bizarre abilities. Plants that can simply draw heavy metals such as nickel from the ground.
We have a really large individual of Pichinendra cutuminata here, which is famous all around the world for containing a blue-green sap that has 25% nickel in it. So I'm going to make a small cut in the tree to exudate some sap that we then can collect and analyze. We should see some sap coming out soon. The green color you can see from the sap is actually the color of nickel. I can use a little bit of test paper to press against it, and we should see a pretty strong color reaction. So the amount of nickel in this sap is between 20 and 25%, which makes it the strangest biological liquid known to man. That is really quite extraordinary to think there's so much metal contained in the sap of a living, healthy tree.
Here in the
Isn't that weird? I mean, this is really high concentration. There are plants like this all across the world. And these are some plants from Malaysia that has really high levels of nickel in the phloem tissue and in the latex. And in also some of these plants are accumulating nickel in the leaf epidermis, on the skin of the leaf, including in the trichomes. These are little hairs that you get with glands at the tip on the leaves, right? So this is a phenomenon that was first observed in the 70s, and then people started studying this in the 80s. And now there are entire companies that are using these plants to clean up metal contaminated soil, which is phytoremediation, and also to extract heavy metals from the soil and sell the heavy metals, and that's called phytomining, right? So there's a really cool, if you're interested in testing this in California, let me know. I'll send you a little recipe that you can make at home. And all you need is some filter paper to look for hyperaccumulators of nickel in California. We only have two. One is where you are, but there must be more. We just haven't looked for it. It's a color reaction that you can use in a simple filter paper crushing experiment that would indicate whether the plant has at least 0.1% nickel. There are lots of plants in the world. I mean, I wouldn't say lots, but some plants in the world that take up weird metals, like even gold and things like cobalt and manganese. Most of the plants that take up heavy metals take up nickel. And is that because we've studied serpentine soils, which nickel is very high in? Or is it because somehow nickel seems to be easier to get into plants than other metals do? But there's a lot of rare earth elements, including some stuff that get into cell phones that plants accumulate. So instead of mining for metals to make cell phones out of, you could potentially use plants to take up metals. So why do you guys think plants take up heavy metals? Why do you want to take up a toxic element? Any ideas?
It might kill the herbivores that ate.
You are right, Carol. It is called the metal defense hypothesis. The idea that plants that grow on serpentine soils are basically using heavy metals in the soil rather than making their own organic chemicals, like some of the organic compounds. They take the metals in, they localize it in the skin of the plant or the phloem tissue, which is sugar rich, or the latex. And then when insects bite into the plant, they get deterred by it and hopefully never return, right? So this has been tested by people like Bob Boyd to showing that this actually is one of the primary reasons that plants may have evolved this weird strategy of making themselves toxic. There are two plants in California. One is called Streptanthus Polygloidis. The other one is what you have, Noscia fenderi, which is also called a Claspi species that is found in Northern California that take up heavy metals. The one on the left here is from South Africa, but both these plants have insects that specialize on this plant. So coevolution is one of those things. You evolve a strategy to keep an insect from eating you, but then the insect would evolve tolerance to it, right? So these are some cool examples of coevolution you can study. Melanocles Boidea is an insect that's found in the foothills of the Sierras, specializing eating only the tissue of Streptanthus polygloidis, which is a California endemic, a serpentine endemics. And in South Africa, you have Berkia cordia in the Asteraceae, which also has a specialized insect that feeds on it, right? So there are a lot of cool studies that can be done, but I wanted to kind of end the talk by telling you that serpentine outcrops are under threat, right? So this is the slide that I showed at the very beginning, taken by my advisor in the 1980s. This is what the outcrop looked like. In 2009, this is what the outcrop looked like. So this is basically, 1980s, this is the 2000s, and it hasn't gotten any better, right? So what's happening with serpentine outcrops, at least in the Bay Area and in places where there are highly, lots of vehicles driving around, a lot of industrial activity, what you find is nitrogen deposition. Aerial nitrogen deposition, wet deposition comes through rain. In the Bay Area, they think that about 11 to 16 kilograms of nitrogen is raining in the Bay Area. So these are, remember I said serpentine outcrops are generally nitrogen poor, and suddenly the nitrogen levels have spiked. And in response to the nitrogen spiking in the serpentine soils, you have all of these non-native grasses that are Eurasian that have now encroached serpentine outcrops. And not only are they impacting the native plant diversity in the Bay Area, but they are also impacting insects that specialize on plants that are endemic to serpentine soils. So the Bay checkerspot butterfly's decline is largely attributed to the decline in the populations of Castanea excerta and Plantego erecta that the Bay checkerspot butterfly's larvae feeds on. So these kinds of changes that are happening across the world are having major influences, impacts on the serpentine biota, not just in California, but also in other parts of the world. Finally, you can conserve serpentine outcrops and at the same time make money out of conservation, right? So this is a huge incentive for conservation in many parts of the world, including in Europe and in Africa, where farmers that were previously trying to get rid of their weeds are now growing those weeds as crops, and they're mining heavy metals out of the soil, right? So here is a farmer that actually met in Albania, and this is a documentary that hasn't come out yet, so you guys are the first to see it, on agro-mining, meaning using serpentine endemics to farm metals.
Only one thing grows everywhere here, yellow tuft. For centuries, the farmers have regarded this inconspicuous yellow plant as nothing but a weed. Now it's the bearer of hope for a better future.
No, we had no idea at all to begin with, because it was something of no value for us. It is something that was of no use for anything. Not even the animals will eat this plant. It was therefore something useless, something with no value.
The farmers here are pioneers. They will be the first farmers in the world to cultivate plants in order to harvest not food, but nickel. Yellow tuft, Alyssum murali. When this plant is harvested and burned, its ashes contain 20% nickel. It thrives in East Albania, especially on so-called serpentine soils or ultramafic soils, which contain a particularly large amount of nickel. French and Albanian scientists have jointly initiated nickel cultivation here.
10% of the surface in Albania is ultramafic site. And we arrive to extract from these serpentine soils more than 100 kilonickel per hectare.
The region around Poiska in East Albania can certainly use the nickel project. The people here don't have much, a little agriculture, but the soils are frequently too poor for growing crops, too much nickel.
All right. Sorry, I don't know what's going on here. Almost there, sorry.
We lost your screen share.
Yeah, I don't know. Yeah, hold on, I got it.
Okay, there we go.
Okay, got it. Yeah. So the point here is that you basically are growing a plant that is endemic to serpentine soils in degraded serpentine soils, not undisturbed serpentine soils. That would be good. But going into areas where people are already trying to farm on serpentine soil, you get them to grow a plant, you extract the above ground biomass, you post-harvest it, and then you end up with a metal-rich bio-ore. So you treat it like a crop. You do watering, you do add fertilizer to the soil, you get them to grow faster, bigger, you get more metal doing that way. So this is called agro-mining now. It used to be called phytomining, but now that in places like Africa, South Australia, and in Europe, people are growing these as crops. It is now called agro-mining. Here's my friend at University of Melbourne holding $70,000 worth of gold, harvested from Brassica jancea, which is a naturalized Brassicaceae plant found across California. It's native to India. It grows on ultramafic soils. It can take up gold on gold tailings, if you treat the soil with a toxic compound, so they're using swimming pools where there's not leaching happening to extract gold out of Brassica jancea. In South Africa, they're using Berkshire codii to extract nickel out of degraded, old, abandoned, serpentine-associated nickel mines across South Africa. There are problems. Arisa murale, the one that you just saw in Albania, was brought to California, to your part of California, by the USDA in the 1980s. I'm sure you guys have heard of this. And if you're interested in this little video, I'm not going to show it because I'm running out of time. It has become an invasive species. And that is a problem. So if you are using agromining, you want to use native plants, not bring plants from other parts of the world where they can then escape and become problems in other places. You're also facilitating the spread of heavy metals from serpentine outcrops to other areas. Soil amendments can be toxic. And then, of course, there's a lot of research going on now, genetically modifying plants like poplars and willows with metal-tolerant genes from serpentine plants for agromining, right? That is a dangerous path to be on, right? Just like we are worried about GMO food, we could potentially end up having to worry about plants that are genetically modified to take up heavy metals for phytomining. Lastly, there are people from about 45 countries that study serpentine soils. We have conferences every 3 to 4 years, just like the mini serpentine conference that Carol put together for you guys. But the cool thing is we get people from all across the world, and we've had conferences every 3 to 4 years, starting from the one in 1991 at UC Davis. The latest one was in Albania and Greece in 2017. And if you're interested in the next conference, and these conferences lead to proceedings where people can publish their research. And if you're interested in attending the next conference, get in touch with me. It's going to happen in Russia, in the Southern Urals. And we get a lot of people from CNPS and California Botanical Society attend these conferences, along with other native plant societies from across the world, because serpentine outcrops are interesting places for people who like plants. And it's going to be at the Ural Federal University, and it's going to be in June of 2022. Hopefully, the pandemic will be under control, and we can get together and talk about serpentine plants at this particular conference. So with that, I want to end. I'm sorry that I went a little over my time limit, but one of the things that I always try to kind of reinforce is that these outcrops, whether they're dumps or pristine serpentine settings, are really model settings to study all sorts of cool biological questions, from very basic sciences to very applied sciences like the phytomining, agromining thing that I just talked about. With that, I'm going to end my talk, and with apologies for going over my time limit, and also with thanks to spending a lot of your time on Sunday evening with me and listening to what I had to say about serpentine plants. Thank you for your attention.
Thank you, Nishi. Yes, that wasn't hard staying with you all that time. Let's see. So if you have questions, put them in the chat, or unmute yourself either by clicking on the mute button, or you can temporarily unmute by pressing on your space bar and holding it down while you talk. That's quicker.
If you just want to talk and ask a question, that'll be fine. I'm happy. I mean, it's a small group, so we can have a conversation.
Yeah. So I worry about all the excitement of mining nickel on these places, because if you plant up all the serpentine with these crops, I mean, it's a crop. What happens to all the diversity that was there before?
I mean, I agree with you, Carol. I mean, it's not something that we have ever promoted, at least me in my writing and my talks, for people to kind of take on. I mean, we were very disturbed when this Arisa murale was brought to California in the 90s. It has now escaped, and it is a problem in serpentine outcrops along the North Coast. And in Oregon, it's an invasive species, right? But it's brought by a company working with the USDA to phytomine. There was a company that started it, and the company went bankrupt, and they did not remove the plants, and the plants escaped. So I feel like if you are going to do phytomining, you've got to do it in places that are already being, used for agriculture, right? So the places in Albania where they're doing phytomining are places where people have been farming for 200-300 years, right? They have been farming in these outcrops that have already been disturbed and cleared up, and not ever making enough money by growing crops in soils that are not good for things like potatoes and corn and soybeans. So for them, it's a big deal because now they're making more money than they've ever made. And it's not really, it's not really impacting the native plant diversity because the native plants have never really grown there in the last 200-300 years, right? So the point is, do you want to restore that landscape to look like a native serpentine outcrop, or do you want people to make some money off that land that they've lived on for 200-300 years, right? So I'm okay with people phytomining in degraded places or places where mining has happened. But I worry that with money, people are greedy, right? There's more money flowing in. People realize that, oh, maybe we need to start encroaching the serpentine outcrop on the other hillside and converting it into a agro-mining setting, right? A farm, a cropland. And that could happen, right? So that could be a problem. But so far, I mean, it's at the early stages of phytomining right now. It hasn't been a big issue. I'm worried about the GMO aspects because people are actually converting poplars that are fast growing and big with genes from the Brassicaceae, which some of these plants that take up heavy metals are in the mustard family. And that can be a problem because we eat a lot of mustards like kale, kohlrabi, broccoli, cabbage, all of those things in the mustard family. And we are going to genetically modify species that can take up more metal. And then what if these genes escape and get into our crop Brassicaceae, right? And that could be then have huge ecosystem, human health implications. So there are problems. So, but at the same time, I think in some settings, like in South Africa, where I've been, where people live in really degraded landscapes, they make very little money. For them, this is a great way to make money and at the same time clean the soil, right? Because phytomining is not going to be permanent, because at some point the nickel in the soil is going to be taken up, right? And that point, phytomining nickel is not going to be profitable, right? So there's always a time limit, but the danger of that is what you are implying. When that happens, will people start moving into other serpentine outcrops that are undisturbed? I don't encourage doing it. I just wanted you to know that this is happening. I would rather have people convert this landscape restore them into what a natural serpentine outcrop looks like in that region. But I mean, there's always that conflict of, what do you do with people who live in marginal landscapes where they have no money and they have to make a living? Is this a better way to make them able to use that land than try to grow crops that are toxic to eat to begin with, because these crops could have heavy metals in them?
Yeah, it's a problem.
It's a problem.
But it is kind of exciting that people have figured out this unusual way of mining that doesn't involve digging a big hole in the ground.
Anything, you're right. I mean, I would always support phytomining over traditional mining, right? I mean, that is an environmental disaster, going into areas and digging up huge pit mines, right? So this is essentially using plants. The problem is that the reason that they're going after poplars now is because poplars have deep roots. These plants that they're using currently are small plants with smaller root systems, so they can only tap into heavy metals in the first foot or two. Whereas if you get a poplar that has, meters long root system, you could suck up the heavy metals at a much larger concentration. And the biomass is such that you can have a huge amount of metal trapped in the biomass. So any kind of recovery is going to be much more energy cost efficient than trying to extract heavy metal from a tiny plant.
Well, in the chats, I'm seeing lots of comments about how really interesting it all was, Nishi. But I guess it's all new to people. They're not coming up with questions.
Well, that's great. I mean, I'm glad you guys realized that. I mean, I just wanted people to I just wanted to share some of these new things that's happening on Serpentine, because in California, we think about Serpentine as our kind of model setting for thinking about diversity and rare plants and conservation. But Serpentine is global, right? It's happening, it's everywhere. And people are studying it everywhere. There's a lot of cool research happening elsewhere. And at the same time, in some of these countries where people are financially strapped, I think they're trying to come up with creative ways for people to make a living, especially in places where they're living next to mine tailings, right? I mean, like gold, like in South Africa and Johannesburg, I mean, the entire city of Johannesburg is sitting on a gold mine, right? I mean, these are mining countries where they've had 200-300 years of mining that has destroyed the landscape. So in those kinds of places where there's so much surface contamination, mining can be a useful way to think about contamination. Elizabeth wants to know about calcium. Yeah, calcium, it's a nutrient, right? So plants actually have a lot of calcium in the tissue to begin with. Serpentine plants don't have a lot of calcium because calcium is lacking in the soil. And that's one of the stresses that serpentine plants have, how they can the fact that they can live on serpentine soil with limited amounts of calcium is a problem. But I don't think we'll ever get to that point where people are going to start mining calcium by using plants, because, I don't think it's going to be I don't think it's going to be very cost effective for something like calcium, because it's not, nickel is expensive, right? You can sell it, right? Where calcium, is more, I guess, it would be great if you don't mine limestone, right? If you can use plants to do it, but plants have a ton of calcium to begin with. It's not like in nickel, where most plants don't have nickel in the tissue. Only about 500 species in the world has high levels of nickel in it. So when you come to phytomining, I think people always take into account the cost effectiveness of the practice, right? Can you actually make enough money to make it worth it? And with nickel and gold and zinc and copper and some of these rare earth elements that go into our cell phones, I'm all for cell phone stuff. These cell phones have a lot of nasty metals in them, right? And we get rid of these cell phones every few years, and it just goes into a dump, and it's just contaminating the environment. And if anybody wants to recycle these elements that are used in cell phones and electronics, I'm all for it. I think there's a lot of abusive mining practices that lead to rare earth element mining, and there are plants that actually can take up some of these metals. And in those kinds of settings, I think you just kind of look at the cost benefit associated with the operation, and you got to make a decision that's best for the environment and also best for the people who live in those areas.
I have a question.
Yeah, so Elizabeth, I don't know who Elizabeth is. Elizabeth McGee from San Jose State? I know an Elizabeth McGee from San Jose State, but probably not. Yeah, calcium is the biggest problem that serpentine plants have to deal with, because there's no calcium or limited amount of calcium in the serpentine soil, right? So plants have to deal with limited amounts of calcium, and the way they do that is they have really efficient calcium uptake systems where they can take up calcium from limited amounts of calcium in the soil. So they have really cool physiological ways to extract calcium from the soil, yeah, so I'm not quite sure exactly where you're going with that question, Elizabeth, but calcium is a big problem for serpentine plants.
I guess what I was actually, I was going to bring this up is that I kind of know that incense cedar will accumulate or take in calcium, and a friend and I were on a camping trip, and he is those lichens, and he said, "Oh, there's a lichen that just lives on dead incense cedar," and so we were hypothesizing, oh, is this lichen breaking down the needles of the incense cedar and adding the calcium that it then will take up, because you notice we just noticed that the incense cedar does well, and the needles just accumulate and decompose at the base of the incense cedar. I just wondered if anyone was doing any studies on that or anything.
That's a cool observation, Elizabeth. I have not heard of that, like in terms of the lichen that grows on dead branches of incense cedar. So this is Calocedrus.
Yes. Yeah.
Yeah, I have not seen it, but I'm sure there could be. But I mean, the levels of calcium, plants somehow take up the calciums, whether the calcium gets I don't really know. I mean, calcium is such an important component of the plant cell.
For sure, yeah.
So it's every plant has calcium in it. And whether the calcium is somehow kind of, the lichen is helping recycle the calcium back into, because any kind of decomposition of plant material that goes back into serpentine soil will add the calcium back to the soil, right?
Sure, yeah.
In fact, one of the hypotheses, I know Carol pointed out that the heavy metal reason is for deterring insects, which is the primary hypothesis, but there's another hypothesis called elemental allelopathy. And that basically says that the plants that are taking up heavy metals are basically localizing them in the bark and in the leaves and the fruits, and then all of these things drop onto the bottom of the plant, and that through decay will release the heavy metals and make the soil right next to the plant really toxic, so other seeds cannot germinate.
Exactly. Yeah.
That's another hypothesis that I'm really interested in testing. There have been a few studies in New Caledonia on it, that people have shown that plants are less likely to germinate under a nickel hyperaccumulating plant. But yeah, I mean, that kind of recycling, kind of biogeochemistry, there's a lot that we don't know much about. Yeah.
That'd be kind of a fun thing to study.
Yeah. I mean, it's a gold mine. I mean, like, any system you look at, there's so many unanswered questions than the answered questions, right? So that's a cool thing about being a teacher, that you can get your students anytime. Like now, I'm going to think about the question that you brought up, and I'm going to try to see whether anybody is interested in looking into something. I have like Michael, the lichenology student. I have another student studying a rare lichen that grows in Los Osos. We are trying to get it listed as the 3rd federally endangered lichen in the US. And it's not on serpentine, but it's on coastal kind of dune habitat called Sarkaria Acidifera. And it's a lichen that grows, it's a splitting yarn lichen that is only found in Los Osos, that only found on a bunch of different shrubs. And so I have a bunch of students who are interested in lichens, and I'll run this by them to see whether they have heard anything about the.
Yeah, that'd be great. I'd be curious, and yeah.
Awesome. Yeah, do you remember the name of the lichen?
No, I'd have to ask my friend, but I could send it to you.
Yeah, just email it. Email me.
I will.
Yeah, that's great.
I'll email you. Yeah, for sure.
Thank you.
Yeah.
Well, it's 8:30, Nishi, and we might not be able to cover all of plant physiology and serpentine biology and everything this evening. Maybe we should let people go home.
Yep, I agree.
Yeah, including you. But it was a wonderful evening. Golly, we jumped around the whole world, and we learned about new things, and we certainly have a deeper appreciation for our serpentine outcrops here.
Great. Well, thank you. And if you're going to do this again next year in person, I'll be happy to join you.
Well, I'll remember that. Thank you.
All right. Well, have a good night, everybody. Thank you again for having me for a talk.
Okay. Good night, Nishi. And
thank you. Good night.
Good night, everyone.
Good night.
Let's see.